Thank you. All right, cool. So good morning, everyone. I'm really happy to be here to present my research. And this is actually based off some of my PhD stuff. So my third PhD chapter, and it's basically comparing how past climate or humans might have impacted the population expansion of a really common lowland Fijian bee species. I'm also happy to see that there are quite a few Australians presenting here, and I apologise to those as I've now betrayed them for the Northern Hemisphere and Yale Uni. Cool. Okay, so just for context, for those who aren't that aware, these are the bees that I was working with, so the Homalictus bees. For you guys in the Northern Hemisphere, it's mostly a subgenus of Iseoglossum. So they're pretty small, between 3 and 5 millimeters long, and they can get up to maybe about 8 millimeters sometimes. I'm sorry to say that I still don't understand the imperial measurement system, but for all of you playing along at home, they're pretty little. They nest in the ground and they pollinate a variety of different plants. A lot of them are generalists, and they can also come in a variety of colors, as you can see here, which I think are quite pleasing to the eye. So they're found throughout Australia, the Pacific, and as far north as southern China and India. All right, so of course I'm working on the Fijian Homalictus, but this phylogeny here is just to show that they're actually the result of a single colonization event from the rest of the Pacific. And this phylogeny sadly only shows described species from Fiji, of which 9 are described in my thesis. But as you can see, the taxon sampling across the broader area is pretty thin. So it'd be really nice to get more sequences from the Pacific as well as Asia to try to figure out actually what's going on there. But zooming in a bit more just to the Fijian clade, so it's really quite diverse. We have 22 species identified from this paper using CO1 data and male genitalic morphology. So of course, the age-old adage of there being a bee's dick difference, in this case between species, is actually really quite true. On top of that, you can see that most of these species are actually restricted to above 800 meters above sea level in the Fijian cloud forests. Sadly, most of this highland diversity is supercryptic, which is why we're using things like CO1 and genitalic morphology to tell them apart. So while those highland species are super interesting and why they're so diverse, that's actually a topic of a completely different talk. So sadly, we're just talking about this one super abundant lowland species, Homalictus fijiensis, which I agree is not a very creative name, but it's much less misleading than the Australian species Hylaeus fijiensis, which is a tale as to why you should get your labels correct. Anyway, with so many sequences, we thought that we had a pretty good opportunity here to look at their genetics and maybe infer what's happened in the past. And if maybe past climate has driven changes in population size, like has previously been inferred from another study, or if it was something else. All right, so I'll quickly touch on my methods. So we have a whole bunch of collections and a whole bunch of CO1 sequences from across most of the archipelago. And we also have some single nucleotide polymorphism data. So we basically have a bit of both so that we can try and compare the patterns that we get with mitochondrial DNA and genomic DNA, because of course mitochondrial DNA can be a bit of a problem. So for the CO1 analyses, we did some pretty simple haplotype networks and mismatch analyses, as well as a bit more complex extended Bayesian skyline plot, which is the EPSP here. And we also tried to, for those more sensitive analyses, use only the third codon position, which is a bit more neutral. For the SNP analyses, we just did extended Bayesian skyline plots and this horrible DIY ABC-RF, which stands for do-it-yourself approximate Bayesian computation with random forest machine learning. And I think you understand now why they made it into an acronym. Finally, we also did some geographic information system stuff, basically to map how the islands might have looked with past climate change, particularly island size and connectivity. So what did we find? Well, to start with, we have sequences from these islands that are labeled and colored here. But we have most sequences from the main island of Viti Levu in black, and the fourth largest island of Kundavu in red. We can also see that in the past, so this yellow here is about 11,500 years ago, the archipelago would have been about twice the size and much better connected between the islands. But for now, let's pay particular attention to the black and the red as I bring up this horrible haplotype network. Cool. So first of all, of course, you can see there's lots of black and red splotches as well as some other things. So each one of these spots represents a unique haplotype, and the dashes between them is a mutation event. So the first thing you might notice is that for Viti Levu, there's, well, a whole bunch of haplotypes, but there's only one mutation normally between each haplotype. If you compare this to the smaller island of Kondavu, you can see that normally there's a whole bunch more mutations between. So this is kind of our first indication that the two islands have had different pasts in terms of demography. And on top of that, you can see that Kondavu actually links to a lot of these south and eastern islands, which is maybe not what you'd expect by least distance. You might expect Viti Levu to be most connected. Cool. So we started to investigate this, like I said before, with simple mismatch diagrams. So here the red lines indicate the actual observed number of nucleotide differences, whereas the blue line is the expected under sudden population expansion. So for Viti Levu, you can see that the observed matches the expected quite well. So it seems like they may have undergone a population expansion event, whereas Kondavu has all of these extra peaks and troughs. So again, this is just showing that something else has probably happened on Kondavu, but it doesn't really answer our question yet as to what happened and why. So for that, we used extended Bayesian skyline plots with effective population size on the y-axis and time in thousands of years along the x. So you can see for Viti Levu that at about 3,000 years ago, the bees went a bit bunter and population started to increase quite sharply, whereas for Kondavu, they've maintained like a relatively stable population size for the past 11,500 years. And just so you know, these red lines, basically beyond those, we can't really infer effective population size. We just don't have the data for it, even though the plot likes to put it there anyway. Anyway, we can confirm that these two islands have had very different demographic histories. But why? Just quickly, these are the EBSB results for our SNP data across different levels of filtering. Sadly, we don't have these data for Kondavu, only for Viti Levu, but the broad trend of population increase is supported. Okay, so just to put this into context, first of all, I'll simply overlay these two EBSB plots on top of each other so that they share the same axis. Now, if I overlay these with some simple climate data from across the rest of the tropics, so the middle graph here is sea surface temperature over the past from now to 15,000 years ago and relative sea level across the same time. You can see that across the past 11,500 years ago, sea surface temperature in the tropics hasn't really changed that much. So we kind of think that it's unlikely that temperature change has driven the population increase. But relative sea level in the past 11,500 years ago, 11,500 years has actually halved the island's area, but certainly not in the past 3,000 years. So it seems unlikely that climate or sea level change has actually driven any population changes in the species on these two islands. So the question then is, what about humans? So if you watch here, if we overlay when humans arrived, it was about 3,000 years ago, and in the next 1,300 years, they ramp up their impact on the environment. So there was a post-Lapita settlement period, as well as first really good evidence of slash and burn agriculture about 1,700 years ago. And of course, you can see that these coincide really well with the increase in population size on Viti Levu. The question is then, well, one, why would human habitation increase population sizes for this bee? And two, why didn't this happen on Kondavu? So first of all, we know that the Fijian Homalictus, and particularly Homalictus fijiensis, is a super generalist pollinator in Fiji. They go to a broader diversity of plants than even the archetypal super generalist Apis mellifera. And they also like to nest in the ground. So, big patches of sunny bed earth will actually probably be quite good for them. So we kind of predicted that the species should be able to take advantage of wide-scale disturbance, invasive species, and probably changes even in plant communities. Secondly, so Kondavu here has a much less history of human habitation. It's still relatively intact. And this is probably because Kindavu is quite steep and has rocky terrain, and it also has a very recent past of volcanic activity, which is actually captured by some of the stories that the Fijians tell. So we think that Kindavu is kind of acting as a control for human alteration of the habitat in comparison to Viti Levu. So that's kind of neat. So I'll wrap things up now. Our broad conclusions are basically that there is population structure in the species across the islands, and that's why we need to analyze the different islands independently. On Viti Levu, the population size has increased while it's remained stable on Kindavu. And the genomic DNA and the mitochondrial DNA about match for Viti Levu. So that's just checking an assumption, which is good. And on Viti Levu, the population increase coincided with humans instead of climate. Whereas for Kindavu, they've remained stable for about the past 11,500 years, and we're kind of taking this as a control for minimal human alteration of the environment. Great. Why do we care about all of this? So first of all, it shows that humans can have an impact on bees, which many of you know is probably kind of hard to prove, and it's that they have so in the past. These impacts can also be positive, which is maybe unexpected, but there are always winners and losers when habitats and environments change. We can also infer really very recent demographic changes. And this is cool, I think, because we can start to tease apart more broadly the relative contributions of humans versus climate. And this is a hotly debated topic in Australia and the rest of the world, of course, when it comes to relative impacts of humans on megafaunal extinctions. And also I highlight the need for assumption checking to make sure that, you know, things such as panmixia and neutrality are taken into account before drawing conclusions. So I'd like to very quickly thank all the people who helped me collect specimens in Fiji, including some of these lovely people here, but there are many more, as well as my funding bodies and the bees for willingly donating their bodies to science and making the project possible. Thank you.
Cool. That was a very cool talk. So there's about 3 minutes for questions. If anyone wants to drop them in the question and answer box or raise your hand and I can read your question for you. Oh, it looks like Amro has a question.
Hello, thanks so much for the fabulous talk. I'm just wondering how sensitive is are the inference methods to kind of your priors?
It depends on the priors. So we actually used the mutation rate from C. elegans for this. So we did some, actually, why do I have the extra slide here? No, I don't. We compared between different mutation rates and between C. elegans and Drosophila melanogaster, for example. And it didn't make a huge amount of difference as to which one we chose. C. elegans actually showed the later population increase, like the furthest into the past. So I think that no matter which one we use, we're still going to be relatively robust as in terms of lining it up with humans. On top of that, we also analyzed 3, 4, and 5 generations per year. So 4 is what we actually use, which is what's been said from other authors. But either way, they still kind of all line up. So hopefully that answers your question. That the most sensitive thing that I'm worried about is probably mutation rates or generation times. Yeah, I think they're fine.
Thank you so much. Yeah, that answered my question. Thank you.
Okay, great. Thanks, Amro.
So Clement also has a question.
Yeah, I don't know whether you can hear me.
I
The
can.
diagram that showed multiple mutations between haplotypes in Kindavu was interesting. Could you just go into a little more detail about why you think that happened?
Yeah, sure. So Kindavu is this really weird island for this species. So I kind of worried in the past that potentially there are multiple species here because of all of these like extra mutations. But I don't really think that's the case because they're kind of spread out all over the place and they link quite closely with these other islands. And like the genitalic morphology basically looks the same. But it's basically suggesting that they've had a longer history of occupation and like relative stability on Kadavu, because they've been able to accumulate those mutations through time. That's compared to Viti Levu, which it appears, the reason it seems like there's a sudden population increase is because all of the haplotypes are really closely related. So they've probably had maybe some founder effect, basically, where if habitat has been cleared, they've just kind of gone crazy and one has spread across. Yeah, so that's why we were also trying to compare it with our nuclear DNA, because we're also worried that potentially this could have been due to a Wolbachia sweep, sweeping across the island and changing the demographic patterns on Viti Levu. But do you have any specific questions besides that about the habitat network?
I'm just wondering, can you tell if there's a smaller effective population size on Kadavu? I think that may have been in one of your slides, but it went by fast.
As in, if there was
As compared to
population
Viti Levu.
size?
Because if you have more mutations accumulating by drift in a smaller population size per unit of time, then you would expect more transitions between haplotypes.
I'm not sure. I'll have to think about this. Maybe we can talk about this a little bit later.
Okay.
Unless you're talking about effective population size or the relative differences of those.
Okay, thanks.